Welcome students to the third part of biomolecules, the last part. First compound in biomolecules, we have covered carbohydrate. The second compound in biomolecule proteins we have done. And now the third is your nucleic acid. So this nucleic acid is present in the nuclei of all living cells. You know this already. It is long thread-like structure, which is macromolecule, a big molecule of high molecular mass. So it is a polymer. And these nucleic acid play important role in all these 3 points important in the development in living organism, in the reproduction of living organism, and protein synthesis in living organism. So these are the 3 main functions of nucleic acid. These nucleic acids are of 2 kinds, DNA and RNA. You know very well. And structure of DNA and RNA we have to do. So it is having 2 main structure, that is your nucleotide. So nucleic acid is considered as polymer of nucleotide. So nucleic acid is also known as polymer of nucleotide. What is this nucleotide? That you have to know, but not in much detail. So nucleotide is the unit of base. It consists of base and sugar and phosphate. These 3 chemicals constitute nucleotide. You need to know the name. Order also you cannot change. They are linked such a way: base, sugar and phosphate. This base is nitrogen base. In the base, nitrogen is present. Heterocyclic means in the ring. Carbon and hydrogens are there, you know, organic compound. But beside carbon, some of the carbons are replaced by nitrogen. So ring becomes heterocyclic. This corner, carbon atom is not present. Carbon is replaced by nitrogen. And these are heterocyclic nitrogen base. You can say these are heterocyclic nitrogen base, and so they are called derivatives. These are derived of either purine or pyrimidine. You need to know this name. These names are important. These are the 2 bases present in nucleic acids, whether it is DNA or RNA. They are purine and pyrimidine, and structure also you need to know. So you can see that purine consists of this structure: 1 hexagonal ring attached to pentagonal ring, and you can see at 3 places, at position 1, position 3 and position 9. Instead of carbon, a nitrogen atom is there. So you must know the structure. Now the second semester questions will come descriptive, and so you may be asked to draw the structure, to make the structure. So this is of purine, general structure, and this is of pyrimidine. Pyrimidine hexagonal ring, and at 2 places you can see nitrogen. Then another important thing that you need to learn, these names are important to know. Nucleotide is also important in this. Then the 2 kinds of purines are adenine. Some people pronounce it as adenine also. So adenine symbolized by capital A, and second example of purine is guanine, which is symbolized by capital G. And here pyrimidine are of three types, three examples. Thymine, symbolized by capital T. Cytosine, symbolized by capital C. Uracil, symbolized by capital U. So, RNA and DNA when we are talking about, then RNA and DNA altogether they have, RNA has 4 of these purine and pyrimidine. It has 2 purine and 2 pyrimidine. Okay, 2 purine and 2 pyrimidine. 2 purine and 2 pyrimidine will be making DNA and RNA, but the difference is that one of the pyrimidine is different in DNA and RNA. So if you see about the type of purine and pyrimidine present, then this again you have to learn important. Both of them are having adenine, guanine and cytosine. Adenine, guanine and cytosine. RNA has uracil and DNA has thymine. So RNA, so this is present in RNA, this is present in RNA and DNA too. This is also present in DNA and cytosine is also present in RNA and DNA. Now left over are thymine and uracil. So thymine and uracil, they are, one of them is present in DNA. This is present in DNA and this is present in RNA. So this you need to know. And then next thing we'll see the second part of the structure, the chemical building unit, that is your nucleoside. So second structure of nucleic acid is nucleoside. Nucleotide was a base, sugar and phosphate unit. That nucleotide was consist of base, sugar and phosphate. Okay, and nucleoside is having only base and sugar part. So nucleosides are called base sugar unit. Nucleotides are called base plus sugar plus phosphate unit. So nucleic acids have 2 units. One is nucleotide and the other is nucleoside. So nucleosides are, here you can see that which is your base sugar unit. So nucleotide, which is base, sugar and phosphate unit. So actually this nucleotide is repeated over and over again through this part. So base and sugar. So phosphate is the chemical that links one nucleotide. It links nucleotides. One nucleotide with the other nucleotide. So how does it link? That you can see phosphate, this is here is your one nucleotide. This is here another nucleotide. So 2 nucleotides are linked together through phosphate. So phosphate function is to link one nucleotide unit with the other through the sugar. So base is here, this base is your purine and pyrimidine. Then base is attached to sugar. Sugar is attached to phosphate, so this is one nucleotide. And here another nucleotide. So a square bracket shows the chain is very long. Small n is, it may be 100 or thousands molecules of nucleotide. So they are linked together. This bond is showing it is continued still chain, and here also chain is continued. Another nucleotide unit is here, another nucleotide unit is here. So this way they are linked together. This linking you should know. And then this is your nucleic acid. This is the structure of your nucleic acid. Okay, this is called primary structure. Basic structure of a nucleic acid. So this is forming one strand. One strand it is forming, and that strand is not straight, it is coiled like this. Okay, so this is your strand. In this way the base is present, sugar is present in phosphate nucleotide with this. Okay. Now this a single strand which is formed by this union of this nucleotide which is making single strand. This strand is coiling. One strand is coiling against the other like this. So when it is coiling around this, around the same common axis, it is coiling. So that is one, and then it gives it 3D structure, complicated structure, which is given by James Watson and Francis Crick. You should know the name. So DNA molecule has double helix structure, and RNA molecule has single. So this double helical structure is this is single. This is a single. So single structure is of RNA and double structure, standard structure is of DNA. The two right-handed helical polynucleotide, this is polynucleotide, strands coiled together in the form of a double helix. This is important, double helix around the same central axis. Then two strands are anti-parallel. You can see they are not parallel. This is not parallel to this. It is not the one coil this, then the other coil this. So one is this side, other is that side. So anti-parallel. So this is another thing you should know. Two strands are anti-parallel. And these two strands, this point is most important. Two strands are held together by hydrogen bonds, and these hydrogen bonds are present in bases, that is purines and pyrimidine. So hydrogen bond, you know, purine and pyrimidine structure which we saw previously, it has nitrogen in its molecule, that's why it can establish hydrogen bond. So how hydrogen bond is established? That is, if adenine is here and guanine is here in one strand, and then the other is coming in contact such a way, then thymine of this, there are thymine and your cytosine also present. Here also your thymine, cytosine, then adenine and guanine will be there like this. It will be. Okay. So adenine and thymine in one strand, guanine and cytosine of the other strand, and then again your thymine, then cytosine, and again here adenine and guanine like this. These strands are coiled such a way that these groups approach. So when these are approaching, you should know always thymine will be in front of adenine and cytosine will be before guanine. And so what is important here? Two hydrogen bonds will be formed between adenine and thymine. So one strand is held by the other strand because of this hydrogen bonds. So between adenine and thymine, two hydrogen bonds. Between guanine and cytosine, there will be three hydrogen bonds. So this part is very important to know. And so one strand is held by the other strand. So hope this is clear to you. And in RNA we have only single strand. We don't have this hydrogen bonding needed because it is single strand. So full form of DNA you know already, deoxyribonucleic acid, and RNA is ribonucleic acid. So only this much you have to do in nucleic acid. So here ends your syllabus, and so that's all. Prepare. In the book you will find much elaborated form. For knowledge you can read, but for examination point of view, according to the syllabus, only this much you have to do. So that's all.